So that's why if you looked at the course calendar, you'd see that we're going to be on chapter 6 for quite a while. Plus your projects for the semester, hopefully they're coming along well, since they're due in a week. And they do constitute a considerable portion of your course grade. But they have to do with enzymes and the structure function studies of enzymes. And so in order to be able to do it well, then you need to be able to know the terminology and everything that's associated with that. And so chapter 6 really is just a continuation of the last 2 or 3 chapters. It's just this one's now specifically about enzymes, and they're going to focus on proteinaceous enzymes, even though there are some that are nucleic acid based. Okay. So first off, we're just going to have some terminology, and a lot of this part should be review, in the sense that an enzyme is just a biological catalyst. Okay. As I alluded to already, it is primarily going to be protein-based. The vast majority of them are going to be proteins. Now, they could be multiple subunits, or they could be a single polypeptide protein. However, there are some that are RNAs. The self-splicing RNAs, I think your book, the most recent edition, lists them as enzymes. However, technically, that's a ribozyme. It's not an enzyme. The reason being is because its substrate is itself, so it does get changed. And so I don't know why they... Just a little, for people who are enzymologists, they kind of cringe at that fact, because an enzyme, is supposed to be a catalyst, so therefore it shouldn't be changed. But a self-splicing RNA is one that cuts itself out of the transcript. So technically, it's not a true enzyme. But there are some RNAs that are true enzymes. And in particular, these that are ribonucleoprotein complexes, the most famous or infamous of which has to be the ribosome. The chemistry is done by the RNA components, and then the proteins are smaller and are accessories to help with either substrate specificity or product release or something like that. And then the one that I worked on was ribonuclease P, which is a ubiquitous enzyme found in all organisms and even in certain organelles. Okay, and the enzymes, what they do is they're going to greatly increase the rate of the reaction, and we're talking sometimes up to 10 to the power 20 over the uncatalyzed reaction. Right? It should be noted that if it increases the forward rate, it will also increase the backward rate. Okay, so it does not change the equilibrium constant. Since it's not a substrate itself, a true enzyme doesn't change the equilibrium constant. And so that's not how, and we'll talk about how enzymes really work and how that they are efficient in catalysis. There are some that are very specific to where they will only look at one stereoisomer, and then there are others that can do multiple substrates for various reactions. Though the term that they use is called promiscuous. If it's promiscuous, it means it can accept multiple substrates. Okay. So, yeah. And then the rate depends upon the activation energy, and we'll go over the energy diagrams in greater detail in a moment. And so I do expect you to know free energy equations and how enthalpy and entropy and equilibrium constant are related, because that should all be reviewed from Gen Chem and organic chemistry and already this chapter. Okay. All right. Existing. So some of the inorganic ions can be cofactors. I've already mentioned magnesium. And just one thing I will point out right here is the fact that you see, for example, with magnesium, you see this kinase and a kinase. Even potassium says kinase. Kinase, that's the enzyme name for something that transfers phosphates utilizing ATP or GTP. So those will almost always require magnesium. But ACE, A-S-E ending, just in general, indicates an enzyme. Okay? But others that are used, there's copper, and we'll see copper with the metabolic pathway of the electron transport chain next semester in greater detail. Iron, Iron's used, magnesium, molybdenum, nickel, selenium, zinc. Zinc is used in a lot of enzymes, but these are just some of the metal ions that can be used. Okay, do not memorize this table. Just know that they are there and that they are important.
So
Mm-hmm.
you were saying that DNA is highly negative. Is that why all these are positively charged, except for the neutral ones? Like selenium?
Well, no, because not all of those will work on DNA. In fact, I'm looking to see if any of those are DNA enzymes. Ribonucleotide reductase works on ribonucleotide, so usually RNA, but otherwise everything else on that list they have protein subunits.
So are
I mean, they work on protein or they work on electron transport chains or something like that.
So why are most of them positively charged?
Well, so for example, all of those that say oxidase, those are ones that are in redox reactions. And so what do redox reactions? What are they?
It's where like increase oxygen bonds and decrease oxygen bonds.
Okay, even think back to Gen Chem. What about redox?
Electrons?
Those are electrons. Remember electrons, remember the line goes GR or oil rig or whatever. And so even the increasing the carbon to oxygen bonds or decreasing them, that's just another way of saying you're gaining or losing electrons. And so if you're moving electrons around, what's great to have around to help move or accept electrons and give electrons back? It's these especially these transition elements. And so that copper can be between copper 1 and 2, iron between iron 2 and 3. There's some iron 1. But yeah, the ones that are like magnesium and manganese, well, manganese has multiple oxidation states, but magnesium does not. And so it's usually, we see hexokinase, those are with the transfer of phosphates. So technically, hexokinase is a transfer of phosphates from ATP to sugars that contain 6 carbons. Okay. All right. All right. So some enzymes, not all, but some enzymes require what's called a cofactor. And a cofactor, and there's another term called a coenzyme, and I'm just as bad as everybody else. Many times we use these interchangeably. There is a slight nuance. A cofactor is some type of chemical component that's required for enzymatic activity. Usually we're talking about like metal ions or something small. So, hemoglobin's not an enzyme. They're just showing here that iron, how iron is required for oxygen to bind. But a lot of the DNA enzymes will require magnesium. You'll find out the reason why later on. But it should make sense. Magnesium. What charge is magnesium? +2. And then DNA, what charge does DNA have? It's negative. I mean, it's highly negative. So it should make sense that you're going to have positive charges around. And so almost always, later on, whenever we start doing metabolic pathways next semester, any of those that have to do with phosphate transfers or ATP or something like that, you can almost be guaranteed there's going to be a magnesium cofactor. Before, that is really loud. I mean, are they deaf next door? The good news is at least we don't have the same person just playing the piano behind us. Pardon?
It's the American Sign Language class next door.
Okay. And so, as I mentioned, there are cofactors and then there are coenzymes. Coenzymes are complex organic molecules or metallomolecules. They can even, for example, the enzyme that I worked on, RNase P, it had a coenzyme, except we always called it a protein cofactor, but it was a protein. It was a really small protein. But many of these coenzymes are based off of your vitamins. And so you've heard like coenzyme A and your flavin complexes and things like that. So these are some of your vitamins that are your coenzymes. Okay, so, and I do want you to know some of the just broad generalities, because that way you can see what these coenzymes are and know what kind of reactions occurring. Like TPP, how it transfers aldehydes or acyl groups. lipoic acid is going to be the transfer of acyl groups. NADH, FADH2, all of those have to do with redox reactions.
That's so much better. Silence.
Oh, okay. All right. All right, so here we go. So this is from the Leninger books. So we can see here, whoops, I thought I had on. There we go. We see here that coenzyme A is involved in the transfer of acyl groups. Those are like your fatty acyl groups, your fatty acids. It could also be acetyl. Acetyl is just the simplest of the acyl groups because it only has 2 carbons total. And we get that from our diet, with pantothenic acid and some of the other compounds. Then you have a lot of your B vitamins, which are involved. Most of them are going to be involved in the transfer of electrons and redox reactions. However, we have some, like B12, which is a cobalamin-based one, which we'll go into. It's really cool. We'll talk about it in greater detail next semester because it has a very odd mechanism, and it transfers alkyl groups. Okay, and so that one you definitely have to get from your diet. Lipoic acid can transfer electrons, but it's also known for transferring acyl groups at the same time, and it's one that actually you don't have to get from your diet. We can't generate that one. But when we have pyridoxal phosphate, which that one we're going to see a lot of later on, it's the transfer of amino groups. This nicotinamide adenine dinucleotide, that's this NAD plus / NADH. And it's known for the transfer of electrons. So a hydride is just another way of doing that. It transfers electrons and a hydrogen at the same time. And then the flavins, that's FAD, FADH2, and they are also involved in redox reactions. This is TPP, thiamine pyrophosphate at the very end. It's one that does aldehydes. And then tetrahydrofolate comes, of course, from folate or folic acid, and it's involved in the transfer of methyl groups in particular. Okay, which is the one that if you are a female of childbearing years, even if you're not thinking about having children in your, immediately, you should definitely still take a folic acid or a multivitamin that contains folic acid because it's something that you need plenty of well in advance of having children. I also have another. This one, in the old PowerPoint, it won't let me move. It'll let me move the table, but not let me move the background behind it, and so you can't read it otherwise. So it came from the old book, but this is just a broad generalization. It says the exact same thing, and I do not know why pyridoxal phosphate and the nicotinamide is in a red box. So your guess is as good as mine. But it says the exact same thing. It's just worded a little differently. Okay, finally we have some additional terminology here, and that's holoenzyme, apoenzyme, and we've already talked about prosthetic groups. And so I'm going to start with the prosthetic group first, because remember, it was the example that we had before. What was the prosthetic group that we talked about in the past? The heme. And so a prosthetic group is usually covalently attached, or it's KD, the dissociation constant, which we'll talk about what that means in a little bit. Its KD is so small that it's essentially not going to be able to pry it apart. So like an antibody with an antigen. Okay, I mean, those are so complementary to each other and they're bound so tightly that it's almost like a covalent bond. Okay. Apo, so here, this is kind of hard, at least kind of hard for me to see it up there. Wow, just hawk up the lung. So, apo, let me just out of the way. Apo, that just means without, or it's lacking something. And so an apoenzyme means it's missing something, like its coenzyme or cofactor, that prohibits it from being catalytically active. Whereas the term holo, not hello, but holo, that means it has everything there in order for it to be catalytically active. So, holoenzyme, for example, when I worked with RNase-P, holoenzyme meant that you had to have the RNA and the protein together. and magnesium. Of course, it's kind of hard to remove all the magnesium from RNA since it's already bound. An apo would be that we had the RNA, but it didn't have the protein. But, or for example, we had people in our lab that worked on zinc enzyme carbonic anhydrase, for example, and it required zinc, and so it was considered the apo enzyme if it didn't have the zinc with it. You had to have the zinc in order for it to be the holoenzyme. There is a board. It's the Enzyme Commission, I think is the technical term, but just like there's the IUPAC for naming the organic molecules, there is one that officially names all enzymes. And a couple slides back, there's one that said that it did transamination reactions, which sounds really cool, but that's not the official term anymore. Now they're called aminotransferases because they transfer amino groups. But this classification, the Enzyme Commission classification, they give each enzyme sort of like a social security number. It's a number that's specific. It will start with a number 1 through 6 because there are 6 families of enzymes, and then they'll say like dot, and they'll give like another number and a dot and another number and so on. And that's because each of those groups of numbers refer to like a subfamily and such. You don't need to memorize all the subfamilies and everything, but it is nice to know what the broad groups, and I would expect what the 6 broad groups do. Okay. Now, there are some later on that their common names get carried over. However, those usually will make sense. Like if you notice, nuclease isn't up here, but what do nucleases do? Those are enzymes that cut nucleic acids. Proteases aren't up here, but those are enzymes that cut proteins. And so, but the broad general classes are oxidoreductases, which those are involved in redox reactions. So we're talking about the movement of electrons. Okay. Transferases, which are the easiest because they transfer something. So that's why aminotransferases specifically transfer amino groups. Amidotransferases would transfer amides. Methyltransferases transfer methyl groups, so on and so forth. Then we have hydrolases. Hydrolases utilize water. And so usually we're transferring something to a water molecule rather than from within. Okay. Lyases, which are the ones that usually people, including myself, forget the most or the easiest. Those have to usually do with the double bonds. You can either form a double bond or break a double bond. Okay, so when all else fails and can't figure anything else, it seems to be the one that it's going to be like make more sense next semester with doing metabolic pathways because you'll see like the same thing over and over and over again. And once in a while you get some enzyme that you just can't really figure out what it's doing, and it'll be a lyase. Isomerases, those just go from one isomer to another isomer. So for example, maybe it goes from 2-phosphoglycerate to make it a 3-phosphoglycerate, where the phosphate literally just went from one carbon to the next carbon. That would be an isomerase. There are some other common names, like a mutase is like the common name for an isomerase, because it makes mutants.
So the transferase always transfers to something besides the molecule?
Right. It goes from one thing to another thing completely. Whereas the isomerase, it transfers it within the same compound, the same molecule. You're just changing from one isomer to another isomer. So the transferase that we'll see the most of probably, especially next semester, are the aminotransferases, and that will literally take it from an amino acid and transfer it to what's called an alpha keto acid. And so it turns a ketone into an amino group. Or the one that took it from went from an amino group to a ketone. So that's why it's a ping pong reaction. And that is the official term for it. It's called ping pong. And then we have the ligases, which you guys probably, what ligase are you the most familiar with? DNA ligase. And I mean, and what happens here is we're forming these single bonds, do some type of, condensation reactions, and it's going to be usually coupled to ATP or GTP, for that matter. There may be some that do like CTP or UTP, but the most common is ATP. And it doesn't have to be DNA or RNA. It can be just something that works with carbon-carbon bonds, as you can see here, carbon-sulfur, carbon-oxygen, and carbon-nitrogen. Those are the 6 broad families. And then, as you'll find out, like there are kinases, phosphorylases, phosphatases. Those all have to do with phosphates. But those are going to be subcategories or common names for some of these. And you're going to get to where, if you know the enzyme name, you can usually figure out, you got a good chance of figuring out basically what it does. Or if you can see the action of what it does, you can come up with a logical guess of what its name is. And so, like, let's see, off the top of my head, aspartate aminotransferase is going to be transferring amino groups to or from aspartate. And so, yep. All right. Okay, so this should once again be a review. The nice thing about looking at things in vivo is that we can assume a relative constant pressure and temperature. There's not going to be a huge difference. Yes, we have some biological portions of our body that may be warmer or cooler, but it's not by orders of magnitude. And so that makes the thermodynamics more simple for us here because of the fact that we can assume, standard body temperature, standard pressure. Okay, and so therefore we can get this delta G is equal to delta H minus T delta S. And I would ask what those would review with you what those are, but the answer is given already, so I assume you can read. But the one thing here that I wanted to point out is if you think back to Gen Chem or organic for that matter, and we see that delta G naught, the little zero looking thing, the degree sign, what did that indicate? Pardon? Say it with some conviction.
State function?
No, not in this instance. What does it indicate for reactions for chemistry and organic chemistry? Standard conditions. And so what are the standard conditions in Gen Chem or organic? Do you remember? Like what's the standard temperature? 25 degrees Celsius, which was, 298 degrees Kelvin. And pressure was 1 atm. And let's see, what about concentration? That's one that people usually forget. What's standard concentration? 1 molar. Okay, so what would be the problem with using those tables in vivo?
Your body's not at any of that.
Right. I mean, the 1 atm is not too bad, but the temperature of the body is what?
37.
37. Okay, so it's 37 degrees Celsius is the same in Kelvin as
298
it's, what's 298 plus 12? It's 310. It's 310 Kelvin. Okay, what's the problem with saying 1 molar?
It's not always going to be.
I mean, what do you have in your body that's 1 molar concentration? Like, what's pH? If it's 1 molar of protons, can you imagine like what the pH of that would be? It's extremely, extremely, extremely acidic, like more acidic than your stomach. And so we use different standard conditions, and that's what this little symbol down here means. A lot of times you'll see this delta G naught prime or delta G prime in the naught sign, because that means under biological standard conditions, which will be 37 degrees Celsius, which is 310 Kelvin, it means pH 7, and it means that magnesium concentrations have to be, this is one that I always get confused, it's either 1 millimolar or 5 millimolar, which aren't drastically different than each other, but that, you have to have millimolar concentrations of magnesium. Okay. And so, which should make more sense considering the fact that those are the conditions that we're working at in vivo. So that's why if you see some tables will actually give multiple delta G's, they'll say like delta G, prime, or not, I should say, in delta G not prime or something like that. And that's what that second column would be indicating. Okay, whoops. Let me see. Why did it do that? And we lost our signal. So if we're going to add in the enzyme, let me go back here and just draw, once again, the generic, which you may want to be able to put it all on one. So, and in the generic, we still have the S going to form a product. But once we have an enzyme, even the simplest of mechanisms, it's going to look something like this. And I'm going to put it in this other color as well. We're going to have the enzyme has to bind one substrate at least. So we have some type of binding event. And by the way, that complex here's got a special name. I'll talk about it later. But we're going to call it the ES complex, or the enzyme substrate complex. And we're going to say that's got another bound intermediate where it's formed the product. And of course, then finally we have to get our enzyme back. Okay. Now, each of these intermediates, and this is where some people get confused, each intermediate has to have its own little valley on that bike ride. Okay? And so just by looking at this, we don't know what the rate limiting step is at this point. However, we still know that we start at the same beginning and we have to end at the same spot. And so once again, there has to be some type of activation energy. La 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 la. And a valley. And so that valley is where the ES complex is going to reside. Then we have to have another, oops, that's supposed to connect. Another little hill and a valley for EP. And then we go down. Of course, we're going to have another little hill. And so this is where we get ES. It resides in that valley. And that one resides in this one. Now, if this is the true, if this was a true energy diagram, which I forgot to put that on there. And so here's G. Which step, in the way that I have it drawn, is going to be the rate limiting step going forward? The ES to EP. And you know that because of why? It's the biggest, it's this thing, I always think about it riding my bicycle, it's the biggest hill in that trip. And so it's going to be if you just look at the sheer magnitude from here to here, it's a pretty large hill. But then from this valley to this hill is the largest one. And then we have another little hill, and then you make your product. And so the way that this is drawn, this is the rate limiting step. It doesn't always have to be the rate limiting step. Because, going backward, which is going to be the rate limiting step going backward? Pardon? Close. E plus P to making EP, because the way that this is drawn, this is the biggest hill to go up. So that's why you don't have to have the same rate limiting step going in both directions. I just wanted to point that out. So
You're counting off based on like the valley to the top, right?
right. Mm-hmm. You can also make an assumption here that the EP formation is more stable than the ES formation for the way it's drawn, and that's because it's a lower valley than ES. That's not always necessarily true. Or you may even have more than 2 intermediates. What if there's like 3 or 4 intermediates? Each intermediate is going to have its own valley. Just like, what's this region up here called? At the top of the hill. That's a transition state. So each, going from valley to valley, each one's going to have its own transition state. And that transition state, you can tell what it's going to look more like based off of which valley it's closest to. So for example, if we're at this first transition state, and this, if you remember, this is the symbol for transition state. This is a little double dagger. I'm going to call it transition state 1. Well, this would be step 1, step 2, step 3. Transition state number 1 is going to look more like ES than E plus S. And we know that because the top of the hill is closer to this valley than the first valley. That's actually really important if you're trying to make like an inhibitor for drugs or something like that. Because what's called transition state theory, you want your drug or your inhibitor to look more like the transition state. Because then it kind of gets stuck. The enzyme would get stuck. And the same thing is true for just transporter proteins. It's going to get stuck. And if we look at the second transition state, which one is it going to look a little bit more like? A little more like ES or a little bit more like EP? A little more like ES, and the third one's going to look a little bit more like EP. So, but that's why these diagrams can tell you so much. So here, I'm going to do another one just for practice for you, enzyme diagram. And it's only going to have an ES and an EP for the intermediates as well. Okay. And one thing that can also be true is you can have, especially with the promiscuous enzymes, they can have different rate limiting steps depending upon what reaction that they're performing. So here's G. So on this one, it's a little bit more difficult to tell. You'd almost have to have numbers. But most likely, which is going to be the rate limiting step?
Forward?
Yeah, going forward, I'm sorry.
That second valley to the second peak.
So you're saying from here to here? Or from here to here?
Yeah, the last one.
And so, I mean, quite possibly, once again, I'd have to this one, because I can't draw free-handed very well. But yeah, if this is the biggest hill to climb up going forward, then that would be the rate limiting step. These are really, really close. Whereas if I do it, let me just do a different color with something. Let's say it has another. Whoops, it stopped it. Whoops. I'm having some issues here drawing it. Okay. Where the one that's in the darker color, it's the first step, which, by the way, is called substrate binding. The binding step, why, thank you. The binding step there, in that instance, would be the rate limiting step. And so that's why I would expect for you to, if I give you a diagram and ask you to know which to label it, and so you know that here, I'm going to use the dark one because it's what I have. At the beginning, it's always E plus S. This first, and that way, that first energy well would be the ES complex. The second energy well would be the EP complex. And at the end, it's E plus P. And so, or if it's kind of hard to do here, if I describe the mechanism to you, that you would be able to draw the energy diagram, where if I said draw one, and sometimes there's more than one right answer if you do it that way. If I said, draw an example of where product release, which is that final hump, is rate limiting. And so then you would have to know that that biggest hill going forward would have to be that very last one. It has to be the biggest hill, which is more similar to what I had in the lighter color.
What did you call it?
Pardon? Oh, here, so here, why don't we just do it real quick? All right, this one I want to actually draw out by hand first.